Does precipitation play a major factor in the presence of fecal indicator bacteria? Is there a positive correlation between precipitation and presence of fecal contamination in Austin creeks? In order to determine this, various creek samples were collected on different days by different groups and stored in a lab incubator for 24 hours in order to allow colonies of fecal indicator bacteria to develop. Our results showed that there is in fact a positive correlation between precipitation and fecal indicator bacteria present in the different water samples. The city of Austin, Texas is a highly urbanized city in the United States with a very large sewage system. It stands as the 11th most populous city in the U.S. and the 4th most populous city in the state of Texas. Austin is home to many waterways, rivers, creeks, and lakes, including Colorado River, Lake Travis, and Barton Springs. Furthermore, Austin gets an average of 35 inches of rain annually. My hypothesis is if there is more precipitation, then there will be more fecal contamination present in creeks. Studying the amount of water pollution in bodies such as creeks is very important, as it allows us to investigate potential pathogens and fecal indicator bacteria present in the water. We can use indicator bacteria to be able to estimate the level of fecal contamination in bodies of water. For the most part, these bacteria are not necessarily harmful or dangerous to humans. However, they can be used to indicate the presence of the potential health risks. There are various methods to detect these types of bacteria, including membrane filtration and culture in selective media, which was actually our used method for this experiment. Fecal indicator bacteria are cultured on media, which actually inhibits the growth of other organisms and allows for the growth of species of interest. Various creek samples, in our case, were collected and placed on selective agars, which were then left unbothered for 24 to 48 hours, allowing bacterial colonies to grow. Creek water samples were collected at different locations around the UT campus and placed in an ice chest. Back in the lab, we then made 2 milliliters of a 1/10 solution of PBS solution using an Eppendorf tube in order to be able to dilute the bacterial solutions. We use the PBS as it acts as a buffer, which maintains a very stable pH for bacteria. Afterwards, we used 3 Petri films and labeled each with bacterial concentration of 1, 1/2, and 1/10. After the experiment, viewing the data we collected, it can be determined that there is in fact a positive correlation between precipitation and the amount of fecal contamination present in creeks. It is likely that we got a few outliers in our data due to having collected our samples in such a big group and possibly causing some sort of human error, such as having had a slightly longer trip back to the lab and the ice not keeping the samples at the most ideal temperature. The water samples collected only 1 day after precipitation had a larger presence of fecal indicator bacteria than did the samples collected 2 days after precipitation. Looking at the combined data, we can clearly see that the fecal indicator bacteria collected only 1 day after precipitation was significantly larger than the data collected for 2 days after precipitation. The numbers 1 through 11 on the x-axis represent the different water locations from which we collected our water samples. The results yielded were not unexpected. It was very likely that a correlation among the location and the presence of fecal contamination would have some type of relationship. There is plenty of previous research that suggests that precipitation is in fact a contributing factor in the presence of fecal indicator bacteria. There were of course some outliers, but this could have been caused by many potential factors such as human error or some natural variation. I feel confident enough to say that the hypothesis that if there is more precipitation, then there will be less fecal contamination present in creeks can be eliminated. My results yielded are very similar to those of studies conducted by researchers in the past. In order to potentially expand or further clarify my findings, the amount of water creek samples collected could be increased. Looking at the results yielded from previous research, it can be confidently determined that there is a very strong correlation among the presence of fecal indicator bacteria and the amount of precipitation. In order to assess the microbiological quality of water, we use fecal indicator bacteria. Like previously mentioned, fecal indicator bacteria is not typically disease-causing. However, it is associated with the potential presence of different waterborne pathogens found in fecal contamination. Fecal material coming from warm-blooded animals can contain various microorganisms, such as bacteria and viruses, that can be pathogenic to humans and cause significant health implications. In order to assess the sanitary quality of water and the possible health risks caused by gastrointestinal pathogens, different bacteriological tests for specific indicator bacteria are conducted. It is actually believed that fecal indicator bacteria cannot thrive or live in natural environments since they are adapted to live in the gastrointestinal tract of organisms. Different factors such as temperature, sunlight, or competition with bacteria found naturally in the water can influence the survival of fecal indicator bacteria in the water. Several studies have shown that fecal indicator bacteria survive from a few hours up to maybe several days in water, but they may not survive for days or months in sediments, where they may be protected from things such as predators or sunlight. The survival time for fecal indicator bacteria in water bodies is a function of many environmental influences, and there is no number that applies to all water bodies or even to all times of the year for a single body of water. We simply assume that pathogens die at the same rate as fecal indicator bacteria. Thus, if we find relatively high numbers of fecal indicator bacteria in the environment, we assume that there is an increased likelihood of pathogens being present as well. Fecal indicator bacteria can come from different sources, such as stormwater runoff, which carries animal and bird droppings, or wastewaters from sewage treatment plants. It is very interesting to identify natural precipitation as a contributing factor in the growth of fecal indicator bacteria, as it allows us to better understand these microorganisms and provides us with better insight as to how to deal with sanitation issues in the city of Austin.